Good afternoon, everyone. My name is Claire-Marie Dhaenens, so I'm a molecular biologist in the Lille University Hospital, and I'm pleased to start this retinal dystrophy session today and to present you the new technologies used in for the diagnosis of inherited retinal dystrophies. So, as you know, IRD, so in short for inherited retinal dystrophies, are a large group of heterogeneous disorders classified based on the retinal cells affected at first and based on the clinical presentation. However, a clinical overlap is observed and can make the diagnosis not easy. And this clinical overlap is mainly explained by a genetic overlap. For now, over 270 genes have been found to cause IRD. And as you can see, for a single group, many genes can be responsible, and some of them can be involved in different distinct diseases. And this high heterogeneity was at first a constraint for a genetic diagnosis of IRD. Indeed, in recent past, genes can only been analyzed one by one using very low sensitive scanning methods. Later, Genome sequencing was used as a gold standard, but only coding regions could be analyzed, and this strategy was very time-consuming and few samples could be analyzed at the same time. Fortunately, in the last decade, next-generation sequencing, in short NGS, was developed and highly improved the molecular diagnosis for IRD because it was really adapted to such genetically heterogeneous disorders. And its high capacity enables the analysis of several patients for several genes at the same time, and all in a very accurate manner. So different types of analysis can be performed by NGS, the first one being the sequencing of a panel of genes. It's the most used strategy, especially in France, for a lab diagnosis. Many genes can be analyzed at the same time. One can choose the genes he wants. And interestingly, all types of variants can be detected more or less accurately according to the bioinformatic pipeline sensitivity and characteristics. And for instance, we can find single nucleotide variation in coding region, flanking introns, and also targeting non-coding region, as well as copy number variants that are large deletion or large duplication. The second type of analysis is the whole exome sequencing, in short, WES. This strategy is very interesting because you can analyze at the same time all the homing genes when you use a ready-to-use exome kit, which ones are cost-effective, so you can identify new IRD genes. The counterpart is that only coding regions and flanking introns are investigated, so you cannot detect non-coding regions or and structural variation are maybe quite difficult to analyze. The major problem of these two strategies is the growing evidence for the involvement of intonic variants. I will take the example of ABCA4, for which in a recent study, we detected after a complete sequencing of a gene, variation in introns. These introns are in fact the non-canonical splice sites or deep introns. And we detected also variation in another type of variation that are structural variants. All of these represent 20% of a variant, meaning that a large part of unsolved patients carry a variation in this unexplored region. So to counterpart this problem, other strategies are now possible. A complete sequencing of the genome, named whole genome sequencing, or the complete sequencing of a single gene. In whole genome sequencing, all types of variants can be detected, except large inversion or insertions, and you can therefore find novel IRD genes, but it's very expensive and quite difficult to analyze. The other type of novel strategy is the sequencing of a complete gene. You can choose your favorite gene, for example, and you can analyze it very extensively. That means that you can analyze the exons, the introns, as well as regulating region. So it's quite an easy way to find variations in introns. So from our experience as a routine diagnosis lab, what did we learn from NGS? So since 2014, date of the first use of the NGS in the lab, our strategy evolved a lot, not only in terms of enrichment methods, sequencer, size of the panels, number of panels, and bioinformatic pipelines. It may seem like an advantage because we regularly improve our detection yield, but in fact it was rather a waste of time because we have to change regularly and to validate the method. So now we need a more stable method. Since recently, we have only one large panel comprising more than 200 of genes. And when working on a sequencer with a high capacity, like the Illumina NovaSeq, we are able to analyze 184 patients in the same run every 2 months. This allows us to reduce our turnaround time to less than 12 months. It's quite long, but we have always around 1,000 of patients on the waiting list. Our performance are quite good because it depends on the pathology and on the gene, of course, and it ranges from 30 to 80%. For example, for Stargardt disease, achromatopsia, retinoschisis, we have a positive rate of more than 80%. For Leber amaurosis, congenital stationary night blindness, we are around 70%. Concerning retinitis pigmentosa, it depends according on the mode of inheritance, but it's between 50 and 66%. However, when the indications is less characterized, like maculopathy or retinal dystrophy, the rate is quite low. We are about 30%. So to summarize, the advantage of panels are numerous because we can analyze single case and we do not need a trio. I could say that panels are easy to use. The analysis is quite fast, and we can analyze and write the report for several patients in a single day. It's about 10 and 20. After filtering, we have not so much variant to check. Moreover, we use two complementary bioinformatic pipelines, and we can download the BAM files on a software called Alamut. And this strategy allows us to detect and to improve our detection yield. More, we can quite easily detect copy number variants. And after a bioinformatic input, we can have quality data which are required for the diagnostic purpose. So panels are suitable for diagnosis lab because the positive rate is quite good, and it's also suitable for IRD because we have not so much genes in these disorders. The counterpart is the panel composition should be assessed regularly, and we cannot investigate unknown deep intronic variant. And it's a very important missing thing. What about whole exome sequencing? Whole exome sequencing is not yet developed in France compared to other countries, so we are quite in late. In the lab, use of WES is in progress, mainly to replace panels in coming months, mostly for other indications like intellectual disabilities, for example. And the use of WES is mainly to improve the global lab workflow. That means that we can use only one pipeline for all disorders, and this could lead to reduce the cost. it's not well defined yet if WES will be used for IRD in the lab, because as I said before, we cannot have a non-deep intonic variant, and we cannot have a non-deep intonic variant. So I'm afraid that the detection yield will not be as good as we have actually with the panels. So another option is to dedicate the WES to syndromic or atypical phenotypes. What about our experience in whole genome sequencing? For now, we performed 50 whole genome sequencing for 23 families, always after for unsolved patient who has benefited for a first negative large panel. That means that the patients were either they have either no variant at all, or they carry one variant in a candidate gene. So in total, we found 8 positive cases. Here are the details. So we, among the 23 families, we found no variant at all in 19. We found one variant in 4 patients. And among the 19 patients with no variant, we detected the causal variant in 5 of them. 3 of these variants were coding. That means that we could have found them in WES, and 2 of them were deep intonic variants. Concerning the 4 patients with 1 variant detected in a candidate gene, we found the mutation in for 3 of them, 2 being deep intonic variants, and 1 of the variants was a large deletion in the region upstream the gene, so in the regulatory region. So the weight of the positive weight of detection was around 38%. So the conclusion for this genome sequencing is that we are we really appreciate the use of whole genome sequencing that really improve our lab performance, despite quite disappointing results. But this tool is very perfect to find a second event, mostly a deep intonic variant in a candidate gene. It's also perfect to find an exonic variant in a gene not present in a panel, but this could be used also, could be found in a WES. We can find large deletion breakpoints with in a very accurate manner, and we can detect also structural variant and intergenic regions events. The counterpart is whole genome sequencing is only feasible with trios or with several siblings. Otherwise, it's not possible to analyze the results. The analysis is very fastidious. The storage of the data is another big issue. And the CNV analysis is not yet easy, but maybe it will be improved very soon. And some region of the ORF15 could not be sequenced for now. Another point is the cost is very high. And at last, I would say that it's not possible for us to write a negative report because it's negative for now at the stage of knowledge, but and according to the filtering capacities, but this kind of analysis required to be reassessed regularly. So I would say that whole genome sequencing is not yet suitable for a first line and on a large scale in a routine lab. Oh, sorry. Another strategy is the whole gene sequencing. And different enrichment methods can be used, but I can talk about one of the most performing one. It's the SNPs-based sequencing. And this strategy was performed in the Franz Kramer's lab, where I work in 2019. So the aim of the team was to develop a cost-effective sequencing method based on SNPs. So SNPs is for single molecular inversion probes. And these probes are target 110 base pair of genomic DNA, and more than 3,000 of SNPs were designed to cover the entire ABCA4 gene. So all 50 exons were covered, all introns, as well as upstream and downstream regions. So as you can see, SNPs are overlapping and they can target both strands, one SNP on the plus strand and one SNP on the minus strands. So it's a very specific method, and you can analyze several, up to 200 of patients per run in a very cost-effective manner. It's around 30 euro per sample. The analysis has the advantage to present a high coverage, and all types of variants can be detected. We have no problem of heavy data storage. You have no problem of incidental findings. And I could add also that it's a good option for panel analysis also, whether to use expensive commercial kits, but it requires a more technical developments, but it's maybe not suitable for diagnosis because we have not so much technical time to dedicate to this development. And another inconvenience is that it's feasible for frequent gene in which you have a large number of patients to analyze in the same time. So again, it's quite less suitable for routine diagnosis lab. Here are the different costs of the strategies I mentioned before. So as you can see, for gene panel or for exome sequencing, the costs seem to be similar. But we have to keep in mind that for whole exome sequencing, a trio analysis is recommended, and we have also to consider that for some patients, different panels can be done consecutively. So finally, the price for a gene panel or for an exome is quite the same. On the contrary, whole genome is 10 times more expensive. And as I said before, for targeted gene analysis, if you use SNPs, it's quite very cost effective. As a conclusion, I would say that for a diagnosis lab, in which several hundreds and up to thousands of patients are analyzed per year, the use of panels are very faster and easier than the use of whole exome or whole genome sequencing, because panels present a good yield for IRD diagnosis. WES is also a valuable improvement, but does not investigate deep intrinsic variant, so it's a big issue. So for now, it's my own opinion, for me the best model is that in first intent, large panel with targeted deep intrinsic variant in frequent genes like ABCA4, USH2A is the best option. And in a second tier, the use of whole genome sequencing for the negative patients. And in fact, our major constraint is that we have too many patients to analyze, and we are not yet in capacities to analyze all of them by whole genome sequencing, because we are too few molecular biologists. And we need a fast method to send more results for the patients. But I'm sure we will succeed, and in several years we could afford this whole genome sequencing as a first tier. And when we will do this, we can use a three-step analysis. For example, in the first time, we could analyze in silico large panel, and in the second step, we can analyze the introns of the candidate genes. And if we still not find no variant, we can search for other genes. So I would conclude for a positive note. I would say that we are not yet ready for large-scale whole genome sequencing, but I'm sure in several years we will reach this goal and we will succeed. So I would say I can propose to see you in a while to discuss again this point of view. So I thank you for your attention, and I thank also my team.
Okay, thank you very much, Claire-Marie, for this very nice overview of the different approaches we can use for molecular diagnosis and research. I have a first question on the chat. So for the SMIPS, I don't know if I pronounce this correctly. Do you have any idea of the limit of the number of the genes that could be analyzed at once? This is a question from Patrick Calvas.
No, I think it depends of the capacity of your sequencer. You can analyze several hundreds of genes for sure. If you analyze more genes, you can reduce the number of patients on your world, I think.
Okay, I have a comment from Hélène Dolphus that there's a national effort now in France to deploy whole genome sequencing, and I think that in very short time it will bring very nice results. So
yeah, no, I'm sure also.
I think it's going to be very powerful in the coming months.
Yeah, so that's why I would like to end with a positive slide, because I know for now we are not able to do whole genome sequencing, but for sure in several years we can do it. But we need to improve our analysis first.
Okay, maybe Hélène, you want to comment on that again?
Yes, just there is a national effort with the Plan France Médecine Génomique to carry, in fact, for retinal dystrophy, in the first intent whole genome analysis with in silico panels at first, as you just said. So most of our centers of reference in France are making the effort now to send to the national platforms, and it's starting. So we have not got yet the results, and we know we are lacking enough professionals, molecular biologists in the hospital. So that I totally agree with the point of Claire-Marie. But I think as we have a lot of European attendees today, we should really insist on the fact that France has taken the step on the genomic medicine, and there is today an access for inherited retinal dystrophies to whole genome as in first intent analysis. So maybe we are, and Claire-Marie explained very well, we are now having a very transitional step with panels still ongoing, but in a few months, hopefully, panels will be in silico panels. This is the aim of the general aim that is given by the French Ministry of Health and of Research for the Plan France Médecine Génomique. There are a lot of discussions on the place of panels, on current panels and the future in silico panels. So it's a matter of a lot of discussion. So thank you very much, Claire-Marie, because I think you have given a lot of information, and there is a lot of discussion, but I think we can carry on.